Hello and thank you for joining us. Today's webinar, Genetic Testing in Ophthalmology, Focus on Eye Panels at PreventionGenetics, will be presented by Dr. Madhu Pantrangi. Dr. Pantrangi is a human molecular geneticist at PreventionGenetics in Marshfield, Wisconsin. She received her PhD in genetics from the University of Delhi and Master's in Biotechnology from Pondicherry University. She joined PreventionGenetics in January 2013. Please note that we will not be taking audio questions. If you would like to submit a question, please submit it through either the Q&A or chat dialog box. On the right-hand side of your screen, please feel free to submit questions at any time. All questions will be answered during the Q&A portion of our program after the presentation. At this time, I will hand it over to Dr. Pantrangi.
Thanks for the introduction, Rachel. Welcome everyone to the webinar and thanks for joining us today. Today's webinar topic is genetic testing in ophthalmology, and we will focus on eye panels at PreventionGenetics and also talk about a few cases in this webinar. By the end of this webinar, we hope that you all will be able to explain the importance of genetic testing in ophthalmology and also discuss different testing options for hereditary eye disorders at PreventionGenetics, and also describe the benefits of testing panels versus single gene test for heterogeneous eye disorders, and also discuss about emerging novel therapies that target specific genes and specific variants. Before going to the webinar topic, I would briefly talk about PreventionGenetics, which was founded in 2004 by Dr. James Weber. This is located in Marshfield, Wisconsin, which is in the upper Midwest region of US. PreventionGenetics is a CAP/CLIA and ISO accredited lab, and it's a comprehensive source for high-value clinical germline genetic testing. And we have a largest test menu options in the US, and we offer next-gen sequencing or Sanger sequencing for over 1,800 genes. And we also offer deletion or duplication testing via RSGGH or MLPA for over 1,500 genes. And we also have a chromosomal microarray for detecting any chromosomal aberrations. Recently, we have launched whole exome sequencing named PG Exome, and we also offer DNA banking services. We only take orders from physicians, genetic counselors, or other healthcare professionals, and we do not offer direct-to-consumer testing. Talking about the webinar topic, vision is considered to be the most important out of 5 senses, and visual impairment has been the major health problem worldwide. And according to World Health Organization and American Academy of Ophthalmology, there are approximately 39 million people around the world are blind, and another 246 million people are visually impaired. The major causes of visual impairment are uncorrected refractive errors, followed by cataracts. But if you look at the graph here, on y-axis, it's the percent of blind population versus the different eye disorders. And according to this data, the major or the primary cause of blindness is cataracts, and you can see it accounts approximately 51%, which is followed by uncorrected refractive errors, glaucoma, age-related macular degeneration, etc. And according to the WHO and AAO, approximately 80% of the eye disorders, they can be prevented or cured, or the progression of the disease could be slowed down if they are detected at early stages. And given these statistics, the importance of early and accurate diagnosis cannot be understated, which could be done through genetic testing. And also with the advent of gene therapy and other types of treatments, the identification of patients' genetic information is becoming increasingly important. And the based on what part of the eye has defect, the eye disorders are categorized into two major types. One is anterior segment disorders and the retinal disorders. And the anterior segment disorders are those that has defect in the anterior part of the eye, for example, lens, cornea, etc. And there are some syndromic eye disorders that has significant ocular manifestations, for example, Marfan syndrome, Fabry disease, etc. And aniridia, Axenfeld-Rieger syndrome, all these come under the anterior segment disorders. And the retinal disorders are those that affect the retina, which is at the back of the eye. And the retinal disorders, for example, achromatopsia, Leber congenital amaurosis, retinitis pigmentosa. And the vitreoretinal disorders are those that affect the vitreous humor, which is inside the eye, the liquid present inside the eye, and also the retina is also affected in these disorders, for example, retinoschisis, familial exudative retinopathy. And the disorders that affect the optic nerve are those optic atrophy, Wolfram syndrome, etc. And the clinical diagnosis of the ocular disorders are done based on age of onset, whether it's a congenital disorder or whether it's an early onset or whether it's an age-related disorder. And also based on whether it's an isolated disorder or syndromic disorder, whether it's only confined to the eye or it has other systems are also involved. And also based on whether it's a stationary or progressive disorder. If it is progressive, it is a slowly progressive or a rapidly progressing disorder. And also, as we have seen in the previous slide, what part of the eye has defect. And also based on fundus appearance. You can see here the normal fundus, how it looks, and this is the retinitis pigmentosa affected eye. You can see the characteristic feature of retinitis pigmentosa, which is bone spicules around the peripheral region of the retina. And for the Stargardt disease, the characteristic feature is the yellowish flux in the central region of the retina. However, for the Leber congenital amaurosis, which is a severe retinal disorder, and it accounts with the majority of the childhood blindness. And if you look at the fundus, it has a wide variability based on what type of gene is dysfunctioning. So in these kind of cases, genetic testing would be very helpful for the accurate diagnosis. And also, based on the mode of inheritance, ocular disorders have all sorts of inheritance pattern, autosomal dominant, autosomal recessive, digenic inheritance, X-linked inheritance, and also complex inheritance. So, as I mentioned, the genetic testing would be very helpful in making the accurate diagnosis and also the prognosis. What to expect in the family, or it also helps in making the crucial decisions related to profession, etc. And also it helps in knowing the risk to your future offsprings and also knowing the risk to your family members. Not only that, it also helps the clinicians to advise any treatment options or any clinical trials they might want to advise on. For example, there is a project called Project 3000, which is led by Dr. Bud Tucker from Iowa lab. And their main goal is to genotype all the patients affected by Leber congenital amaurosis, which is estimated to be 3,000 in US. So they want to genotype and populate the patients who have pathogenic variants in RPE65. So, so far I will be talking about the RPE65 gene therapy in my last few slides. So it's been very successful so far, and they're also working on pluripotent stem cells. So now testing may lead to specific treatments in future. Of course, there are disadvantages as well. Earlier testing would make a person live in a state of anxiety, waiting for the symptoms to become evident, and also future blindness may dominate one's life. All these could may overcome through good genetic counseling. And the approach for the genetic testing is based on whether it's a monogenic disorder or heterogeneous disorder. For example, the monogenic disorders such as Stargardt disease and choroideremia, opting one gene would give you high detection rate. Although there is another gene that has recently been associated with the Stargardt disorder, but ABCA4 is the major causative gene for the Stargardt disease, and it gives you high detection rate. For the heterogeneous disorders, which is due to multiple genes, maybe opting next-gen sequencing panels would give you high detection rate. And we have tested Leber congenital amaurosis and ocular cutaneous albinism, and we've found that the detection rate is higher by opting the next-gen sequencing panels. However, there is a disorder called Retinitis pigmentosa, which is a very complex disorder. Even studying or even testing the 80 gene panel would not give you that high detection rate. There is clinical sensitivity is estimated to be approximately 50 to 60%, and maybe that is due to there are still unidentified genes that are associated with the retinitis pigmentosa. So in my next few slides, I will be talking about two segments of the eye disorders, for example, retinal disorders and the anterior segment disorders, and emphasize on the genetic testing importance. When I talk about the retinal disorder, I'll be talking about the cone degeneration and the rod degeneration. So I thought I'll just briefly go over the retina and its role in visual cycle. So retina is the nerve layer, and it's a nerve layer, and it actually creates nerve impulses those nerve impulses are sent through the optic nerve to the brain. And in this retina, if you see closer, the retina has a region called macula, which has the special light sensory cells, which are cones and rods, collectively known as photoreceptor cells. And in that center of the macula, the region called fovea, which has a high density of cones. The cones are involved in the color vision, also the daylight vision, and also the sharp central vision. The region that is surrounding the fovea is that has high density of rods, which are involved in the dim light vision and the peripheral vision. The process that occurs in these photoreceptor cells are called the visual cycle. In this, there are three important steps, which is retinoid cycle and the phototransduction cascade and the ciliary transport. The retinoid cycle, which is the regeneration or the recycling of the retinoid, which is a derivative of vitamin A. And the phototransduction cascade actually generates the nerve impulses and sends to the synaptic terminal. Both these processes occur at the outer segments of the photoreceptor cells. Here in this picture, it is shown only in the rods because it is extensively studied in the rods. And the proteins that are required for this retinoid cycle and phototransduction cascade are not synthesized in the outer segments. They have to come from the inner segment. So the ciliary proteins that helps in the transportation of these required proteins from the inner segments to the outer segment, which are present in the transition zone of this outer and inner segment. And you can see in all these the visual process, there are several proteins that are involved in this visual cycle. So these proteins encoding genes causes several retinal disorders. You can get the information in the RetNet, which is a retinal network, where you get the information of all the retinal disorders. And so far, there are over approximately 300 loci have been mapped, and over 250 genes have been identified. As I mentioned, because all these genes encoding proteins are involved, causes the retinal disorders, they have a huge phenotypic overlap. And you can see in this graph on the x-axis, it is the cone, or whether the disorder is due to the cone degeneration or rod degeneration. And on the y-axis, it's a progression of the disease with the age. And the Leber congenital amaurosis, which I talked previously, it's a very severe retinal degeneration. It actually involves both cone and rod degeneration, and it reaches to the final stage even at the younger age. And achromatopsia, it's only the cone degeneration, and also you can see it reaches the severe stage at the younger age. For some other disorders like Stargardt disease, cone rod dystrophy, they are relatively slowly progressing. And if you look at these disorders, in the earlier stages, they might look like only cone or rod degeneration, but at the later stages, it might turn like both the rods and cones are degenerated. So for these disorders, genetic testing would be very helpful in making the accurate diagnosis. And we do have several panels recently we have released. Those are congenital stationary night blindness, Leber congenital amaurosis, flecked retinal disorder, degeneration panels, achromatopsia, and retinitis pigmentosa panel. And if you look at this picture, you can see how there is a huge genotypic overlap as well. So we have a very comprehensive panels in the market. We test approximately, we test 80 genes for the retinitis pigmentosa panel, and recently we have released these, but we have already tested quite a few patients for these panels. I would talk about one interesting case for that we tested for the retinitis pigmentosa panel. And in this family, we have got a patient's sample, and the client actually ordered a comprehensive retinitis pigmentosa panel. Mentioning about this panel, we do have this retinitis pigmentosa panel broken down into 3 panels based on their inheritance pattern, autosomal dominant, autosomal recessive, and X-linked panels. But the client ordered the comprehensive retinitis pigmentosa panel with the reflex to the deletion duplication testing. How this works is we do the next-gen sequencing test first, and if we see any inconclusive information, or if we find it as negative, then we will do the deletion duplication testing. If we find a positive sequence finding, we would not do the deletion duplication testing. So here, if you look at this family, the patient's mother and the maternal uncle, they both are diagnosed by RP, and the mother was diagnosed at 46 years, and the maternal uncle was diagnosed with retinitis pigmentosa at 30 years. And the patient and the brother both are affected by retinitis pigmentosa at their early 20s. And looking at this pedigree, it looks like autosomal dominant disorder. Interestingly, when we tested this next-gen sequencing panel, we found a protein truncating variant in the RPGR gene, that too in the ORF15 region, and with the which is documented. This variant is documented causative for X-linked retinitis pigmentosa. Usually, the females who are heterozygous carriers of protein truncating variants in the ORF15 region, they exhibit a range of phenotypes that can vary from asymptomatic to a severe disease that is similar to affected male patients, based on the what type of variant, and also probably due to skewed X inactivation. And We also found variants of uncertain significance in other genes as well, and we have a policy for the variants of uncertain significance. When we find some variants of uncertain significance in the patient, we offer free testing for 2 family members so that it can help us determining the clinical significance of these variants of uncertain significance. So the variant that is found in the previous patient was also reported in another pedigree by Churchill et al. And you can see here, even in this pedigree, it looks like autosomal dominant inheritance. And the patient had this mutation in the RPGR. And in their study, approximately 8.5% of the families thought to have autosomal dominant retinitis pigmentosa, and they ended up with X-linked retinitis pigmentosa. So, which is only possible, the accurate diagnosis would be possible through genetic testing. And these results have substantive implications for calculation of recurrence risk, genetic counseling, and potential treatment options. And talking about the X-linked retinitis pigmentosa, it is majorly caused due to mutations in RPGR and the RP2 gene. XLRP accounts for 15% of the retinitis pigmentosa case, and it's a severe subtype. And the RPGR accounts for 70% of the XLRP cases, and RP2 approximately 20% of the cases. And interestingly, the mutations in the RPGR gene, 70% of those, excuse me, of the and 70% of the pathogenic variants in the RPGR gene are located in the ORF15, which is a highly repetitive region. And you can see here the RPGR, it has 2 isoforms, isoform A and isoform C. The isoform C is the one that is associated with the X-linked retinitis pigmentosa. And isoform C has the larger ORF15 compared to the isoform A, and it is highly rich for G. It's a G-rich region, and it's a highly repetitive region, and it's very hard to sequence it. And we have a special chemistry at PreventionGenetics, and you can see here the coverage for this ORF15 region. We have several overlapping sequence traces that helps us making the accurate variant call. And we have tested several patients for this ORF15 region, and we have identified several novel variants. Probably we will publish these results with de-identified patient information soon. And in this particular patient, you can see here, we have an option for RPGR where we test ORF15 region first with reflex to the remainder of the gene, because as I mentioned, the 70% of the RPGR pathogenic variants are in this one exon, which is ORF15. So majority of the patients have positive sequence finding in the ORF15. And here we have identified in this patient the duplication of 109 nucleotides, and which is undocumented, but it is a frame. It causes a translation frame shift and premature protein termination, and this type of variants are causative for X-linked retinitis pigmentosa. So it is expected to be pathogenic. And here I will talk about the ABCA4 mutations and the importance of genetic testing, because the ABCA4 has allelic and phenotypic heterogeneity, and ABCA4 is the protein that helps in the visual cycle. It's a transporting protein, and different ABCA4 mutations causes different types of disorders. And you can see here, the lesser the ABCA4 activity, earlier ABCA4 is known as ABCR, so the lesser the ABCA4 activity, the severe the disease is. And here, the patient with the normal ABCA4 activity is normal, and if the patient is a carrier for a pathogenic variant, based on the severity of the allele, either the individual would be normal or at risk for age-related macular degeneration. If the patient has both mild alleles, the patient have Stargardt disease. If the patient one mild allele and one severe allele, either they would be having the phenotype of Stargardt disease or cone rod dystrophy. And if the patient has two severe alleles on opposite alleles, they have a severe disorder, retinitis pigmentosa. If you go to the Foundation Fighting Blindness website, you find this information that majority of the eye disorders, for example, age-related macular degeneration or retinitis pigmentosa, usually the clinicians, they recommend the vitamin A supplements. However, if the patient has age-related macular degeneration or retinitis pigmentosa that is due to the causative variants in ABCA4, they should restrain taking the vitamin A, because in the visual cycle, you have seen that the vitamin A derivative retinoid is recycled continuously, and when this retinoid is recycled, that has to be transported through this ATP binding transporter protein, which is encoded by ABCA4 gene. If this protein is not functioning very well, then this vitamin A derivative retinoid accumulated in the outer segments, and it converts into deleterious product called lipofuscin. So to avoid that, the vitamin A supplements not to be taken, and also you should be avoiding some food that is rich in vitamin A, and also avoid the excessive sunlight exposure, because upon sunlight exposure, the retinoid is again recycled, and it causes deleterious effect in the retina. So only it is possible to know whether due to ABCA4 mutations, and also the accurate diagnosis only made through genetic testing, because otherwise you will not know whether the variants, the patient's phenotype is whether it's a Stargardt disease or cone rod dystrophy without the help of genetic testing. And here is another gene, which is also the encoded protein is also involved in the visual process, and it causes the retinal disorder. It actually encodes the rhodopsin, which is important protein in the visual cycle. And in this particular pedigree, this affected individuals were tested in another laboratory, and they found the causative variant, which is at the codon 106. Glycine is substituted by arginine, and this variant causes the autosomal dominant retinitis pigmentosa. So when we got the patient samples, you can see the patients who are circled. The patient samples were tested to PG, and when we tested, was homozygous for this variant, and all these individuals were negative, and even the previous lab also had the same kind of results. But when I looked at the pedigree, and this is surprising that the mother is homozygous, but the patient is not carrying that variant. So which made me think that there might be allele dropout. So our R&D team, they have developed another set of primers, and when we repeated the testing of these individuals again, then we found out that the mother was heterozygous but not homozygous, and all these 3 individuals were negative for this variant, and the patient who tested negative was actually the heterozygous was heterozygous for this variant. So when we reported these results, the client was very happy about the care we have taken to release the accurate results for this family. And there's another case for Refsum disorder. This one, due to the mutations in the PHYH gene, it's actually a syndromic disorder. In this particular family, the patient's sibling was tested in a different laboratory, and she was identified to be homozygous for this particular variant in PHYH gene. So this patient's specimen was sent to PreventionGenetics for target testing for this particular variant. Because both the siblings were affected, so they were expecting the same results from the sibling as well. But when we tested the patient, we found she is heterozygous for that variant, but not homozygous. So we recommended that testing the remainder of the gene with reflex to the deletion duplication testing to find the second causative variant in this patient. So then the patient sent a request asking for testing the remainder of the gene. Then we have identified the second causative variant in this individual. So I just want to emphasize here, when we find a homozygous variant in our lab and when we report the results, we usually recommend testing both the parents to see whether the parents are carriers for this variant, just to rule out or confirm the possibility that the opposite allele is deleted, or whether there is any allele dropout in this particular patient family. Because that same thing happened in the sibling who was tested in another laboratory. There was allele dropout. That's why she was shown to be homozygous for this variant. And now I have talked about the retinal disorders and the importance of genetic testing. And here I will talk about the anterior segment disorders. And the first case is about the cerebrotendinous xanthomatosis, which is a rare autosomal recessive disorder with multi-organ involvement. And the primary symptoms are diarrhea and juvenile cataracts. Those are the earliest clinical manifestations of this CTX disorder. And additional symptoms are tendon xanthomas and multiple progressive neurological symptoms. And this is due to causative variants in CYP27A1. And this gene encodes an enzyme which helps in breaking down the cholesterol. When there is a dysfunction, the mutations in this gene and the enzyme is dysfunctioning, then there will be elevated plasma cholesterol and cholesterol levels accumulated in the brain, lens, tendons, and other tissues. And it is shown that the individuals who are affected by this disorder, if they are treated with the cheno-deoxycholic acid, and when they have earliest symptoms, which are diarrhea and juvenile cataracts, it actually normalizes the cholesterol concentrations and abrogates the progression of the disease. This was studied in 4 family members, 4 individuals from the same family, the 2 siblings who are affected, and they were diagnosed at very early stages, and they were treated with this cheno-deoxycholic acid. At the same time, their uncles who were also affected, but they already had additional symptoms, other organs were also affected, and those 4 individuals treated with this enzyme at the same time. When this was done for 14 years, and the 2 siblings who were treated with in their early stages, they remained asymptomatic for 14 years, but only moderate improvement was seen in their uncles. So this is definitely a case where emphasize the prevention is particularly significant in light of the availability of early genetic diagnosis and the devastating effects of this illness if it is not treated. And this is another disorder which is Axenfeld-Rieger Syndrome, which is the case of digenic inheritance. This is actually autosomal dominant disorder, and 40% of the cases are due to causative variants in PTX2 and the FOXC1 genes. So we have a panel that tests approximately 6 genes, and these two PTX2 and FOXC1 are the members of transcription factor genes, and they are involved in regulating the development of different tissues of the anterior segment of the eye. And both are capable of independently regulating the transcription of the target genes. And but FOXC1 and PTX2, they physically interact with their crucial functional domains on both proteins. And in this family, you can see both the parents are affected, and mother was carrying a mutation in PTX2 gene, and the father was having a mutation in FOXC1 gene. And in their children, the son who inherited only inherited the mutation from FOXC1 gene from the father of the FOXC1 mutation was also affected, but the daughter who inherited both the mutations was severely affected. So this is definitely important, emphasize the importance of genetic testing, because they will be able to know the risk to their future offsprings, and also to know the risk to their family members as well. And not only that, genotype and phenotype correlation studies have shown that the PTX2 mutations have more severe prognosis for the glaucoma as compared to the FOXC1 mutations. And even within the FOXC1 mutations, patients who have duplications in FOXC1 are at higher risk for developing glaucoma. So again, the genetic testing would be helpful in knowing what type of mutations they have and what the phenotype is due to what gene. And this is another case of oculocutaneous albinism. Oculocutaneous albinism is severe hyperpigmentation of the skin, hair, and eyes, and can affect people of all ethnic backgrounds. And approximately 1 in 17,000 people have at least one type of albinism. There are approximately 5 types of oculocutaneous albinism. And upon long-term exposure, these individuals are at higher risk for skin damage and skin cancer. And you can see there is a reduced iris pigmentation, which because of that it loses its translucency, and the light cannot enter through the eye. And this is due to this disorder is majorly due to the mutations in TYR and OCA2 genes. And the TYR gene is very important, essential for the melanin biosynthetic pathway, because it plays important role in the first 2 catalytic steps of the melanin biosynthetic pathway. In this case, I will be talking about 2 commonly seen variants. One is at 402 codon, and the other one is at 192 codon. And you can see here the information about this, the prevalence of this, these 2 variants. It is seen in the codon 402 variant is seen in approximately 28% of the general population, and there are several apparently unaffected individuals who are homozygous for this variant, and you can find that information in the exact database. And similar with the 192 codon 192 variant, which is also seen in approximately 25 to 37% of the general population. And there are several apparently unaffected individuals who are homozygous for this particular variant. And if you look at the pathogenicity of this 192, and it is classified as benign. However, the codon 402 variant has a conflicting interpretations of pathogenicity, and it is classified as risk factor, pathogenic, and association with the normal pigmentation, etc. Why these two variants are in huge debate for its pathogenicity? Because they are hypomorphic alleles. The codon 402 variant reduces the TYR activity by 75%, whereas the codon 192 variant reduces the TYR activity approximately 40%. So the 402 variant has been reported in several publications saying that this is commonly seen in individuals who have only one pathogenic variant and affected by oculocutaneous albinism. Probably in that case, kind of situations, if it might be causative for oculocutaneous albinism. But there are you can see in this particular patient information where the probands, affected probands, parents were carrying the 192 variant and 402 variant in trans with several pathogenic variants in the TYR gene, and they're also carriers for the protein truncating variant, but they are not affected. Why this was, as I mentioned, because it reduces the TYR activity, but in several papers they have talked about even that much reduction in the TYR activity is sufficient for the normal melanin biosynthesis. But these results is not convincing to say that the 402 variant in trans with the pathogenic variant could be causative for disease, because we have seen in this patient population. So we have, since 2014, we have tested several individuals for the oculocutaneous albinism panel that has 13 genes, and the results were not convincing that when we find only single pathogenic variant in trans with the 402 variant could be the major cause, primary cause of the disease. However, there is a publication by Jagirdar et al. Where they have in their patient population, they have seen the wild type is prevalence is 35%, and the 192 prevalence variant prevalence is 32%, and the 402 variant is at 30%. However, the haplotype, which is the 402 variant and the 192 variant are on the same allele, that prevalence is only 1.9%, which is relatively low, frequency is low. And also in their patient population, they have seen two individuals who are homozygous for this haplotype, that is mentioned as a rare double variant. They have seen the severely hypopigmented individuals. There aren't many patients, but only two patients, and both were severely hypopigmented. So their hypothesis is maybe these haplotype may have such deficient enzyme activity that it would complement an albinism allele and manifest as an incomplete albinism, such as oculocutaneous albinism type 1B, because the TYR causes the type 1A and type 1B, and the type 1A there is no TYR activity, whereas in type 1B there is a residual TYR activity. And which is a little convincing that the haplotype in trans with the pathogenic variant could be causative for the disorder oculocutaneous albinism. And we have also looked at our previous patients where we have found both these variants, and we have seen several patients were having only one causative variant. Particularly in this patient, the patient was having the 89 kb deletion in TYR gene, and the patient was having the hemizygous variant of this 192 because the opposite allele was deleted, and the patient was heterozygous for this 402 variant. And probably the parental testing would help us to determine that the 192 and the 402 are on the same allele and they are in trans with the deletion allele. So doing the parental study in all these patients would help determine the clinical significance of this haplotype variant. And interestingly, we have done one family study where we have found one pathogenic variant, and the patient is homozygous for the 192 variant and heterozygous for 402 variant. You can see that the family study, both the parents are, the father is homozygous for the 402 variant, and the mother is homozygous for the 192 variant, and the mother also carries the pathogenic variant in the TYR gene. And the father, upon this family study, we found that the father is a carrier of haplotype. And the patient inherited the haplotype from the father and the causative variant from the mother, and the patient is affected. So this is a little convincing that the haplotype, which is a rare allele, if it's present in trans, the pathogenic variant, that could be causing the ocular cutaneous albinism type 1B. With this, I finish my cases, and I will emphasize I will talk about the existing or the under trial novel therapies. The RPE65 gene therapy. The pathogenic variants in the RPE65 accounts for approximately 8% of the Leber congenital amaurosis cases. And there are other genes, LRAT and RDH12, which are in the same pathway of the visual cycle. They also cause the similar features as the RPE65 associated LCA. And obviously the genetic testing would help in differentiating whether it's RPE65 associated LCA or not. And in this study, they have delivered the adeno-associated virus vector comprising the RPE65 gene subretinally, and they have done the follow-up study within few months, and they have seen significant improvement in visual and retinal function, and maximum improvement was achieved within 6 months after the treatment. And also they have done the follow-up study, 3-year follow-up study, and they have seen that the visual improvement was stable. And also you can find more information at the Spark Therapeutics website, where they have successfully completed the phase 3 clinical trial for the RPE65 gene therapy. And they're also working on the gene therapy for other genes, example CHM. And there are also other labs which are still trying to do a gene therapy for ABCA4, USH2A, and other genes. The problem with the ABCA4 is because of its size compared to RPE65 gene is relatively small, and it's efficiently delivered using the adeno-associated vector. Because this vector was used because it elicits minimal immune response and mediate long-term transgene expression in variety of retinal cells. Whereas the ABCA4 efficiently doing the gene therapy for that big gene would be a little difficult. But the clinical trials, they are still the clinical trials are underway. And I would talk about another interesting therapy by Antisense oligonucleotide nucleotide therapy for Leber congenital amaurosis that is caused by a frequent deep intronic variant in CEP290. You can see here the deep intronic variant actually creates a splice donor site, and it results in a inclusion of cryptic exons between the exon 26 and 27. And They have treated, in this study, they have used the immortalized lymphoblastoid cells from individuals who were carrying the homozygous deep intronic variant, and they have transfected with antisense oligonucleotides, which are a stretch of 20 base pairs, and they are very specific to that treatment. They bind to that region by reverse complementarity, and they make that sequence is not available for any further splicing process. So they have done this study by the dose-dependent manner, and you can see the RT-PCR results were showing that in a dose-dependent manner, they completely got rid of the incorrect Step290 mRNA, and you can see here it's only they completely got rid of that cryptic exon. So probably this kind of treatment could be extended to other deep intronic variants, and other disorders can be successfully treated with this kind of novel therapies. And in future, we might have several other therapies that can target specific genes or a specific specific type of variants. And genetic testing would be helpful in knowing what type of mutation or what type of variant you are carrying, and that would lead to specific gene therapy, specific type of therapies, specific therapies. And with this, I finish my webinar. And thank you for your attention. And if you have any questions, I may take. And also, I want to emphasize on the panels that are coming soon at PreventionGenetics. We will have an inherited retinal dystrophies panel that tests approximately 280 genes. Over 280 genes, and we also have the corneal, soon we will have corneal dystrophies, brittle cornea syndrome, and strabismus syndrome panels released at PreventionGenetics. If you have any general information, if you need any general information, you can contact us to this email ID. And if you have any technical questions, and if you have any comments or suggestions, you can contact me through this email ID. I can take any questions you may have. Thank you.
Okay, just as a reminder, if you'd like to submit a question, please submit it through either the Q&A or chat dialog box on the right-hand side of your screen. We'll go ahead and start with the first question, and that is the RPGR repetitive region included in all of your tests?
Yes. Whenever Okay, that's a very good question. Thank you. Whenever we have this RPGR included in our panels, we have this special chemistry that we would do for any panel whenever we have this RPGR is included in that panel. And we have a special chemistry that we do through Sanger sequencing. Even the next-gen panels, we see there is not a good coverage, and the coverage drop in that region. So we always have this special repetitive region covered by Sanger sequencing in all our next-gen panels, and all our sequence tests wherever we include.
Okay, and then the next question is, are documented causative intronic variants going to be included in the inherited retinal dystrophies panel?
Okay, that's a good question too. Yes, including the UTR untranslated region variants included in our inherited retinal dystrophies panel. We have very recurrently reported deep intronic variants in, for example, Step290 that I talked in my previous slide, and also some ABCA4 deep intronic variants, and also other variants that are documented causative. And we are currently working on this panel. Hopefully, this will be released soon. Thank you.
Okay, that looks like all of our questions, so that is going to conclude our program. If you have any additional questions, please feel free to reach out to us at any time. Thank you again for joining us.
Thank you.